So last time we talked briefly why FIJI is for many people the first choice when it comes to image processing and image analysis. We talked a bit about digital images, about such features like bit depth, color space, lookup tables, and defined what are channel slices and frames in a hyperstack, which is a representation of a three-dimensional image or a timelapse image. And then we also mentioned some resources for image processing in general and for FIJI. And then we got to the topic of image processing as a form of visual improvement of the image. So let's pick up where we ended and talk a bit more about this. So we already last time we saw lookup tables, histogram scaling, and now let's get to some other topics in visual image processing. And let's take again some image. We can take the image which we used last time, which is from the examples, open samples, and we have this called focus series. It's a two-channel image. It's a stack with two channels. Let's first look at the red channel only, or the channel which is shown in red. So if I want to separate the two channels, I can go to color, split channels, and I will get two stacks. One is the green stack, which I closed because I don't want to look at it now. And let's look just at the red one. From the look of it, this might be mitochondria in the cells. And you can see that the image is relatively noisy. So one thing which you might want to do as a part of processing or pre-processing of the image is to do some form of denoising. There are, of course, more advanced form of denoising, but some of the very basic and simple ones can be found here on the process. And for example, you have here prepared this function smooth, which does some form of filtering. And if you look to the FIJI documentation, you might find what exactly it is. But if you want to have a little bit more choice, you can go here to filters. And what these filters do is that they look at each pixel and at the surrounding of the pixel and replace the value of the pixel with a value which takes into account the surroundings. So for example, a mean filter will take a pixel and the neighboring pixels and replace the value in the pixel with a mean of the neighboring pixels. So this is basically something what you can call also as a sliding window averaging. Median filter, which is very popular, does something very similar. It just, instead of using the mean of the neighboring pixels, it replaces the pixel value with the median of the neighboring pixels. And I haven't done any study on comparing these two, but generally what is said in the literature and is that the median filter is slightly better in preserving, because basically what these filters do, they of course smooth the noise, but they also will blur sharp features. So the median filter is said to better preserve sharp edges than the mean. Very similar thing is Gaussian blur, which again does the thing that it replaces the value of each pixel with a value calculated as a Gaussian function, Gaussian fit from the neighboring pixels.
Your voice today very soft. It's soft. Yeah, I hear it.
I still hear someone talking in the background. Please, if you can mute yourself for the benefit of everyone else. I cannot find who's the. I don't think there are. I'm not sure if I can make a co-host in this free version. It might be only in the paid subscription. Let's go back to this image and look at these filters. So let's look and even process filters, and let's look at the median filter. And you can define here the radius, which means how large is the neighborhood of each pixel which you will consider for to calculate the median. It can be defined in pixels, or in case you have for the Gaussian blur, you might also use scaled units if you have if you know the size of the pixels in micrometers. You can preview if you want to tune the radius, so you can use a preview. For example, this looks quite nice. It smooths the noise. If I make this larger, if I exaggerate it, then you see I will lose a lot of information because I will basically smooth out too much. So I think these 2 pixels in this case looks reasonable. So I can accept this. And I can apply this on all the images in the stack. So this was smoothing or denoising. And the other thing which I can do is little bit opposite to it, and that's to sharpen the image. So if I go here to process, there's this function sharpen, which is also a form of filter, but it's a filter which is called unsharpened mask. Now, this name unsharpened mask might sound counterintuitive because it sounds as you want to unsharpen things, but it's actually it's a filter which takes an unsharpened, a smooth version of the image and subtracted it from the image, and by that is removing the any blurred, unsharp features while preserving those which are sharp. If I select this thing, again, I can play with the parameters of it. And try this, and you see what it does. It removes the more blurry features. If I reduce this weight, it will little bit less removal and less highlighting the sharper features. I can, if I make the radius very small, then I might be again creating a lot of speckle noise. I can show you if I make it 1, then I might start creating noise in the image. So I can set this, and I have a processed image. Now, of course, all these filters, you see that they might visually enhance some features, but of course, there is no guarantee that what they do to the image improves the accuracy of the image. They might be introducing artificial distortions to the image. So always be cautious when doing any such types of manipulations, because they might actually do more harm than so they might be good for visual observation of the image, but might not be the right thing to do if you want to do any advanced analysis later on. There are, of course, other bit more sophisticated ways how to process images. Let's just maybe this was not the best example to show the how the unsharp mask or the sharpening works. So you can take another sample, these HeLa cells, where it might be a little bit better. Now, if you want to compare things, or if you, sometimes when you want to test some processing and you don't want to lose what you have done so far, it's a good idea to duplicate the image. So I will do image duplicate. Now I will apply process sharpen. You can see in this case it brought quite perceptible visual improvement of the image. I will close this, and let's go back again to the sample we had before, this Convolution series, and let's look at some more sophisticated way of processing. I again take just the red channel, image, color, split channels. And what I want to do now, so there are, of course, there are better, as I mentioned, there are more sophisticated and better ways how to improve the quality of image than just do some filtering and or sharpening. One of the standards in image processing is deconvolution, which is basically trying to find an image which is because, you know, when you are imaging with a microscope, the image which you capture is the ideal image convolved with a point spread function of the microscope. And if you know, you can estimate the point spread function of the microscope, then you can iteratively try to find out something which is close, an estimate of the ideal image, which is not affected by the, not broadened or by the point spread function. So that's the idea of deconvolution. It is quite computationally demanding procedure, and it requires, especially, it very much depends on good estimate of the point spread function. So in this case, because this is the sample from FIJI, for which I don't have the complete metadata, which will tell me what was the objective used, what was the wavelength, all these data which are needed to estimate the point spread function. Then I cannot do deconvolution. I can do something else, show you there are alternatives to deconvolution, which are using artificial intelligence. So people have trained neural networks by feeding them ideal images and images which are broadened by point spread function. So they created synthetic images, ideal image and image which is broadened by a point spread function, so which simulates how would the image look like when acquired with a microscope. And then they trained a neural network to reconstruct from the image, the simulated image with point spread function, the underlying ideal image. Of course, for each specific problem, for each typical for each type of structures you want to image, ideally you should train the neural network on something which simulates the type of structure that you want to image. Otherwise, again, you have the same problem that you might be creating artificial structures. So let's, I don't have trained neural network for this particular type of images, which I don't even know exactly what it is. I assume it's mitochondria, but it's just my guess. But I can go now to plugins, CSV deep, and I can do use the demo neural networks, the trained one. So they have one which is doing deconvolution. It was trained on images of microtubules. So I can run it. I can use the default settings. And now it will take a while. Let's see if we have any long, maybe taking too much capacity. I want to see if there are any If anyone has any questions, you can ask now. You can ask by microphone, because now until this is over, I probably won't be able to see if you have typed any questions in chat. So if you have anything, you can ask now while we are waiting for this to be processed. If we were doing deconvolution, we couldn't do it because probably that will take us until the end of the session. So this is much faster than deconvolution. We are coming slowly to the end of the process. Okay, here we are. So there are now two channels. One is the reconstructed image. The other channel, this is just some uncertainty image, some error image. So we don't want to. Now, if you look, so you see you have something which looks way much sharper. It might look like you have gained a lot of resolution. Of course, as I told you, it was trained on microtubules. So obviously it will always try to, whatever image you feed it, it will try to find out a network of tubules in it. So you can be cautious when applying any such things, as they might produce, they might easily produce something which is not there in the image. So just to show you how you might, if we go to the same slice, we were the slice 10. So now we can compare it side by side. Now, maybe one more thing to this. So, of course, when you have some three-dimensional image, you might do three-dimensional rendering. You might do orthogonal projections, but there's one other quite, in some cases, quite useful way of representing it, and that is to color code the depth. So you create a projection, but each plane is projected in a different color. Let's have a look at it with this image, for example. So it will be under image, hyperstack, and it is called temporal color code. So it's meant for time lapse, that it will color code different time points in different colors. But you can use it also for a stack. You can try to run it. And of course, so one thing it cannot do, so let's do like image, color, split channels. And we close the green one, which we are not interested in. And then we go to image, hyperstack, temporal color code. And you see that's what when I tried it run in the morning. So I and into this program. Maybe your version of FIJI, maybe you have an older version I have updated today. So maybe you have an older version, maybe it will run for you. But to show you, I have found on FIJI GitHub the code which does the thing. So I can now run it here. Run and so we have here a color-coded image. So, and probably not that insightful in this case, but in some cases it might be quite a nice way how to represent three-dimensional structures. Okay, what else we have? So there's one more thing, background subtraction. So let me open some image. Yes. So let's look at this image, for example. And one thing you can notice here, so you see there are some shades in the background. So the big background is not uniform. So that's a typical situation where we want to subtract background. And there is in FIJI, there is a popular algorithm for doing this, which is called rolling ball algorithm. And it again looks at the neighborhood of each pixel. You can define how large the neighborhood should be, and it finds the average in that the local average, and then it determines what should be the local background and subtracts it. So if we go here, process, subtract background, you can select whether the background should be light or lighter than the features or darker than the features, and then we set the rolling ball radius. You can preview. See, this is a typical artifact. If the radius is not set very well, that it creates this checkered like image. So let's try a different radius. This seems to work better. We could try to fine tune it further. But let's for now accept it. And we get an image where the background is more uniform. And now we can again go back to what we did before and go to process, filters, and apply, for example, median filter. And play further with it. And among the filters, I forgot to mention, there's one which is a little bit more complex than these simple filters, that is called the Kuwahara filter, and that is said to preserve very well edges. You can try to run this. And it's not a big difference because you already did the median filter. Another thing which is sometimes useful, not so much for the visual enhancement of the image, but rather as a pre-processing step, and it's also related to these filters. There's a function find edges. So that will find the places with the largest change in the gray value. For example, if I go try to do this, see that it basically finds the contours of your features. So that sometimes can be useful. For example, sometimes it might be helpful for segmentation. We'll get to segmentation in a short while. Now, one thing you might have noticed, so I was using this here under plugins. I was using this plugin which did the neural network processing. And maybe if you have opened your FIJI, you cannot find it there. So that's, as I talked last time, FIJI is it's very modular in its structure. So you have, when you download FIJI, you have a selection of plugins there. But there are many other plugins which you can find. So if you typically, when people create some new image processing, image analysis algorithm, then they might choose to publish it in the form of an FIJI plugin. So then when you read the paper, you might find there a link where you can download the plugin. And a lot of plugins can be downloaded automatically to FIJI because their developers have set up a FIJI update site, and you can select from which update sites you will be updating your FIJI. So you will be when you update FIJI, you will update the core, the ImageJ core, but you can also update selected plugins. It's of course this modular feature structure of FIJI is great because it allows a lot of people to contribute to FIJI, to add a lot of interesting functionalities. It has its drawbacks because sometimes you will find that you have a plugin which works perfectly well, and then you update your FIJI, and because something changes in FIJI itself, suddenly some of your plugins might stop working until the developer again fixes the issue to be compatible with for the plugin to be compatible with the latest FIJI version. So that's the downside. Okay. And of course, the other thing is that there are you might realize that there are so many plugins, and there's usually for like typical tasks like localization analysis, what a lot of people do, you will find that there are plenty of plugins which do about the same. So the typical joke of coding applies here. Why no one uses my code? Because everyone is busy developing their own. And I will show you now how you can select what plugins should be updated. So if you go here to update, you have to run in the check for updates. Hopefully this won't take long because I have updated today in the morning, so there shouldn't be many new updates in the meantime. So we just have to wait a short while, and then we get to the window where we can manage the update sites. And manage update sites, and here you have a selection of a lot of update sites that each corresponds to one or several plugins which were published by some research group. For example, you saw that I was using this CSB deep. So you see here, you can check that you want to be updating CSB. And that's it. A lot of other. Okay. So next thing which I have here is image tag registration. So this is something which you might have to do when you are acquiring time-lapse images. So sometimes over the time, the microscope stage will drift slightly. So over the time, your image might be slightly shifted. I will show you an example to better explain what I'm talking about. So you see here this image, so probably you don't see much. So if just because the histogram is not well scaled, so what we'll do is adjust brightness contrast. We can run the auto. Now you can see what is there. So this is an image. It's nothing interesting. It's just image of fluorescent peaks. It was acquired to test the stability of the microscope stage over time, doing a long time lapse to see how far does it drift. So if I now run it, you can see that the things have moved a bit. Now I can again use the temporal color code. And this one's, oh, so here it is. So you can see nicely the trajectory which the stage, how the stage has moved. Obviously the beats are not moving, so they all follow the same trajectory. So that, of course, could be an issue if you want to analyze a time-lapse image. So there are, again, number of different plugins which can correct for this. Of course, the ideal way how to correct it is to have fiducial markers. That means something like these beads which are sticking to the cover slip, they are not moving. So I know that whatever trajectory these beads follow, that is the trajectory of the whole stage. Sometimes it's not easy to incorporate fiducial markers into the sample. And if I don't have anything fast moving there, then I can assume that the change between two consecutive frames is relatively small. And if I find such a drift of such a shift of the images, which makes the two consecutive frames as similar as possible, then that would be the correction for the drift. I can use again, and let's say, because this again, this will take ages because there are a lot of frames. So I will create a substack. So what I do is image, duplicate, and I won't duplicate the whole stack. I will do just maybe from 50 to this. And now you can see there is still, of course, some drift. And now I go to plugins. I have to find the one I wanted to use. It's this GBSC suite of plugins from University of Sussex. And utilities, align images. So I select, I want to do this. Here I can define what is the maximum translation. So if it moves more than this, then I won't be able to correct for the drift. In 2 frames, it moves a longer distance. And then I can run it. Let's see if we have any questions. I have, okay, I don't have example of counting axons in optic nerve. I will get to counting objects, but not axons in an optic nerve. This is, of course, we can then later, if you have, for example, such an image, we can then get to this topic, or you can bring it on Friday to NOBIC Imaging Clinic. How long was this recorded? It was an overnight. It was overnight recording, so it was starting in the evening before we went home, and it ended next day morning. So something like from 6 p.m. to 9 a.m. roughly. Now nearly done with this thing. So this is the corrected one. So now to see how we have improved, we can again do the color coding. So this is the original one. And this is the corrected one. So you can see the improvement that we have really corrected for the drift. And next we'll move to a big topic of segmentation. So here we are moving from the processing, from the, let's say, the visual aspects or the preparing images for analysis. So this is, we are already now further down the pipeline where we want to identify objects of interest. So that's already getting us to the analysis part, like where we want to measure objects, count objects, and do such things. So typically, before we do the segmentation, we might want to do some form of pre-processing, such as apply some filters. Let's again look at some examples. Let's use, this is an image of DAPI stained nuclei. I think this, now I'm not sure how I got this image. It's not in the samples. I used this some years ago when I did a similar introduction to FIJI. I think it was somehow in the among the FIJI samples long time ago, and over the time it got lost or something like that. It's definitely not an image which I acquired. It's something I remember getting it somewhere from the sample images, that you won't find it there with the current versions. So we have here some objects, nuclei. So we might want to count these nuclei. We might want to measure how large they are, how measure the fluorescence intensity in the objects, and do such sort of things. So for that, we want to identify where the nuclei are. So that's the segmentation, where we say what is the object and what is the background. And we, of course, might do more complex segmentation where it's not just object versus background, but it might be object, one type of object, second type of object, and so on. And the simplest way of segmentation, simplest form of segmentation is by thresholding. So we simply set some threshold and we say what has higher intensity than the threshold, that's the object. What has lower, that's the background. It's very simple to do, but of course it has a lot of limitations. So to do the thresholding, we go here to the image, adjust threshold, and here there are, and so there are, you see, I haven't adjusted the threshold manually. I have in FIJI used some algorithm to determine the threshold automatically. Of course, there are these sliders which allow me to set the threshold manually. But which is, of course, if I have a single image which I want to segment, that's perfectly fine. I can tune the threshold manually. But if I want to apply, say, looking forward, when we want to develop some automatic processing pipeline, then it's not good to have anything manually set in such a pipeline, because the images which might have slightly different intensities, then manual thresholds will go wrong. But if we are using some automatic algorithm, then within a certain range of images, we will get similar results. So that's the advantage of using automatic thresholds. One thing you may notice is that, of course, you can see that it somehow found the nuclei, but it looks a bit speckled. So maybe I was too fast. I should have done some filtering before. So I will still reset this. And first I go here to process, filters, and maybe do a median filter. This looks okay. So I accept this. And then I go back to image, adjust threshold. And now it looks smoother. There are different algorithms, and in different situations, some algorithms might work better, some might work worse. So some tend to set the threshold lower, some tend to set it higher. You can have a look briefly at ImageJ website. They have here a reference for these different thresholding algorithms. So you can see the same image and different algorithms applied on the same image. And you see that some tend to set the threshold lower, so they segment more objects. Maybe they include some background. Some are setting it too high, so they might be losing some objects. A very popular one is Otsu algorithm, but again, it's not a universal answer. So in some cases, it might work very nicely, but in other situations, it might not be the right choice. Thresholds. So we can try to see for ourselves. You can see Otsu. It's not that much different. Triangle. See that it's taking a bit more. This again is maybe similar to Otsu. Here, because so one thing how these algorithms work is that you can see here the histogram of the image, and it's clearly bimodal histogram. So we have some distribution of pixels which form the background, and then we have some distributions of pixels which corresponds to the objects. And then these algorithms are trying to separate, find an order between these two distributions. So here, because you see the histogram has quite well-defined bimodal structures, then most of the algorithms are giving similar outcomes. In some cases, it might not be so clear-cut, especially if you don't have one type of objects, but you have large distribution of intensities. So you have one type of object which is dimmer, some brighter objects, and then you won't see such nice bimodal distribution, but you might see a very broad distribution which doesn't have a clear separation between the background and the objects. And in such cases, the different algorithms might return very different results. It might be actually sometimes useful. I remember I was at there was one user who came to me, and they had some protein, fluorescently labeled protein, which was present both in the nucleus and in cytosol. And they had control cells, and they had some treatment, and they expected to see different ratio between between the intensity in nucleus and the intensity in cytosol. So I could use two different thresholding algorithms, which allowed me to separate. So one algorithm segmented only the nuclei, and the other segmented the whole cell. So I could separate what is the cytosol and what is the nucleus. So in this case, maybe, I don't know, we can stick with this one, and then we apply. And maybe one thing which, again, I mentioned is a good thing to do. You can make it. We can, in case something goes wrong and we want to go back to this image. We will keep it in store. And now we apply, and we will get an image which is a binary image. So there are just two gray values. Okay, they are white and black. So it's a real black and white image. So the black is 0, the white is in this case 255. Now you may see that there are things which are not that nice. For example, sometimes I have some holes in this object. In this case, they are small. Sometimes I might have bigger holes. So there are here on the binary, there are all sorts of functions which allow me to work with binary images. So you can see, for example, a function fill holes. Very obvious what it does, and you see the holes disappeared. The other thing you might notice is that some of the nuclei, because they were very close, they were touching each other. So then they appear here as a single object. So if I want to count nuclei, I would like to somehow separate these and count them as two separate objects. And for that, I can use something which is called process binary, and it is called watershed. It's using watershed algorithm. So it's like you have in geography, you have watersheds, some mountain ridge, and the water on one side flows to one river and one sea, and on the other side it flows to another sea. So that borrows the name from that idea. And if I use it, it tries to split, and in this case you can see that a reasonably well job separating here the where the nuclei were touching each other. Another thing, there are other functions here on the binary. So these are very standard functions: erode, dilate, open, and close. So what do they do? As the name suggests here, it basically shrinks all the objects. So if I press this, you see everything shrunk. If I do process binary and this will obviously do the very opposite, and everything again expanded. But you may have noticed there was one small dot here, which was so small that when I shrunk everything, this small object disappeared. So now when I do the reverse, the dilation, this object, because it disappeared, it's gone. So you see that it's not always perfectly reversible. And this is exactly here what is called morphological opening, what I have done. I have done erosion followed by dilation. So that's the morphological opening. And that can help me to remove small speckles. On the other hand, the opposite is morphological closure, where I do the two procedures in the opposite order. And what it does, it will remove cracks. So if you can imagine what it will do in this case, it will again undo what the watershed algorithm did. Process, binary, rows. You see, I undid what the watershed did. That's not what I wanted to do. So now you see there are still some small objects which are not nice, but I can deal with them when I do the counting. If I want now to count these AI, let's do again image duplicate to keep this binary image. And what I will do now is analyze particles. And this is the feature which is counting objects. So it allows me to select a range of sizes for the objects. In this case, because I don't have any scaling of the image, so it's all in pixels, or in pixels square in this case. If I had scaled image, it would be in micrometers square. So with more meaning, so units. Sorry, my window is not very soundproof, so now being disturbed by thunder and lightning. Not like a thunder, like a lightning. There is sort of the convolution procedures. And this is so. In this case, if you want to see how large your objects are, you can, for example, draw a region of interest. So I draw a region of interest. This is 50. So this is the diameter of this circle is 50. So let's say I don't want to count anything which is smaller than that. So that would be, if I calculate the 25 times 25 times 5. So I can say that I don't want to have anything smaller than 2000 square pixels. Analyze particles. Again, I could set an upper limit if I want to exclude some very large objects. I can set here limits on circularity. So circularity is basically the ratio of the width to the height. So circularity 1 means circle Low circularity means an object, a rod-shaped object, something which is prolonged. So I can say times 0.8. What will it do? And I have the option to exclude objects on edges, which I might want to do in case I am interested in the size of the object. So then objects on edges, I won't be able to see their whole size. So I don't want to, so whatever is touching edges, I don't want. Here are the results. So you see that here it found some objects. For example, this one didn't pass. Maybe it seems to be larger than the limit, but probably I was too strict in terms of circularity. So I might again redo it. Analyze particles. First, because I had a region of interest selected, so it was looking only in the region of interest, not the whole image. So now you see this image, this object was counted as well. So here I have results. So I have the area of each object. I can have the integrated intensity, which in this case doesn't make sense because they all have 255, so it's the area times 255, so it's not a very meaningful parameter. Here, if I do these measurements, maybe I forgot to mention last time that here under Analyze, Set Measurements, so I can select what all measurements will be displayed. So I can have area, I can have the mean gray value. I can also select that I want to have the standard deviation and all sorts of other statistical or other characteristics. And also it adds each of the objects which it finds, it adds them to something which is called ROI manager, region of interest manager. So it marks each of these objects as a region of interest and then adds them to this region of interest manager. This again, you might not see the importance of it at this moment, but this is very useful when if you are writing macros, if you want to automate your image analysis pipeline, because then you can make a loop where you are looking at each of the regions of each region of interest in this ROI manager, and you can do all sorts of other processing on that particular region of interest. You see that I'm highlighting the different regions. It can be also useful, for example, if you have a multi-channel image, because these regions of interest will then propagate to all channels. So if I use one channel to segment these objects, then I can look at the regions of interest in another channel, for example. So now I have this binary image, and of course you saw that I can, for example, count these objects. I can see their area, but I can't. I don't have any information on what was the fluorescence intensity in those objects, because it's a binary image, so I have lost that information. So what if I am interested not just in the area of these objects, but I'm interested in what is the fluorescence intensity in each of those objects, or average intensity in those objects. I want to compare, for example, different samples and segment them, and then look at the fluorescence intensities in the segmented objects. So how can I do that? I can use, of course, these regions of interest, but I might, if I don't want to look at individual objects, I want to just, let's say, look at the objects, all of them globally, then I can use this binary image as a mask. I will show it to you. That's why I have here the binary image, and I can use it as a mask, and I can take the original image, which is here, and apply the mask in this image. So I will here mask out all the background and only take the structures which I have segmented out. How I can do it? Well, obviously, I can, one obvious way would be to multiply these two images. But first, because here I have 255, so I want to change this to 1. So I go here to process, math, divide, and I divide this image by 255. I lose the segmentation, no, image adjust, brightness, contrast. I can here try to set from 0 to 1. Yes, everything is okay, everything is there. So now if I multiply this image with this image, so I can do it here under process, image calculator. I select DAPI confocal 1 multiply by DAPI confocal. I create a new window, and you see here I have, so here is the original intensity, and here is 0. Now if I do, for example, analyze, measure, I have area of the image, I have mean intensity. Of course, this mean intensity is not very insightful because it's the mean of all pixels, including all these pixels which have 0 value. So this might not be the best way how to do it. So I can do it in a bit better way. Start this, and I can do process. I can do it in a different way, and one way which I like to do it is that instead I divide this image with the mask. So I do process, image calculator, and instead of multiply, I will divide it. And I create a 32-bit result. This why, because in a 32, because here I had 0. So probably you know from mathematics that if you divide anything by 0, it doesn't make much sense, and you should get you will get infinity. That's what it says. It's set to value infinity. And if I do now analyze, measure, so you see I have now I have a different mean value. And I have also the minimum value is now not 0, but it's 74, because the infinity doesn't contribute to the mean value. So I have only the mean value from the segmented objects. So this is something which might be interesting when I want to compare different samples. I segment the objects, and I want to get the mean gray value from these objects. So even if in one image there are more objects, they cover larger area than in the other, still this mean value will be a meaningful parameter. Any questions now that we have talked now about the segmentation? So maybe one more thing to the segmentation. I have prepared one more image for this. And it's this image. So this image, again, you won't find exactly this image when you look into the samples, but it's based on this blobs image. What I basically did, first of all, I inverted the lookup table of this blobs image. I added some noise to it, and I added uneven background. So you see that basically this side of the image the whole image is darker, this side is brighter. And this is one typical situation where simply thresholding will fail. You can try it. Let's see, there's a question. Yes, of course, we can do also segmentation in 3D. You can, if you have a stack, you can then apply thresholds on each frame. But there are also better ways. There are segmentation, segment so there are ways how to segment objects in 3D, but it's of course it's much more I would say difficult than in 2 dimensions, because it's easy to do the segmentation on each slice, but then of course that doesn't mean that I have the optimal segmentation which will take into account the 3-dimensional information. In a way, the 3-dimensional information can be taken into account only if I move away from this pixel-based segmentation. What I did, this thresholding, is something what I can call pixel-based segmentation, because I basically look at each individual pixel at a time and look, is it higher than the threshold? Is it lower than the threshold? And I didn't care about the surroundings of the pixel. if I want to utilize the 3D information, I have to look into using the surroundings. I will get to this now in a short while, because it's quite important. It's a very good question. So if I now try to do the same, I do adjust, threshold, and you see there's no way how I can set, even if I manually try to tweak the threshold, there's no way how I can segment both sides of the image. Obviously, because on this side, the background is higher, has higher intensity than the objects on this side. So thresholding won't work in this case. Even though by eye I can clearly see where the objects are, I need something more smarter approach. So one way is to use local thresholding. Let's say now it's just one brush, and we do again the here filters, medium. Okay. Now, when I was preparing this image, I was not that careful enough. So here it happened that at one stage I got some saturation here. So this will obviously affect the segmentation. So just don't look too much at this very edge. It's not a perfect example that I prepared. So yeah, I want to do local threshold, and here you see, if I search, there's auto local threshold. It has different algorithms, and it gives you the option to try all of them. So I will try all of them to see which one works the best. And you see that some of them work quite nice, some of them not that nice, but obviously it's way much better than when we did the global threshold. So for example, this one here failed miserably, but this one, this Bernson's algorithm, seemed to do quite well. Or this mid-gray is also not that bad, or Otsu, local Otsu algorithm also did quite well. So then if I can use this to select which algorithm to use then. So let's say the mid-gray looked quite good. So now I do the auto local threshold, and I will select mid-gray, and I have the segmentation. As I said here, it's a bit messed up by having some saturated pixel during when I was preparing this sample image. Let's go back and let's look at another way how I can segment this type of images, which is another way which is taking into account the surroundings of each pixel. And it's even in a more sophisticated way than the local. So the local threshold, obviously, it was doing some sort of sliding window where it looked at each part of the image and was doing the thresholding algorithm not on the whole image, but found it only for a part of the image. I can do something even more sophisticated. I can use machine learning approaches, which allow me not just to use the intensity information, but they can also use, for example, criteria like the shapes of the objects, all can be taken into account when doing the segmentation. there is, for example, one plugin which is called Trainable Vika segmentation, which can be also used for 2D images as well as 3D images. And now I first need to train the classifier. So that means I will need to draw regions of interest which corresponds to the objects. So I add this to class 1. We draw another one here. Class 1. Obviously, this training is quite a lot of work because I need to here somehow segment. Either I can use some automatics, have some, maybe I could make some image with some automatic segmentation. Otherwise, I have to segment it manually. So of course, if I wanted only to segment a single image, I won't be taking the efforts to train the classifier. I would rather segment it manually if I cannot do it in any better way. And of course, this all makes sense if I want to do an automated procedure. I want to segment thousands of images. So then I take the efforts, I train the classifier, and okay, I won't be dragging it longer. Let's try to train it and see what it does. Obviously, this is not the optimal training, but you see that even with this measly training where I just segmented manually, not very nicely, I didn't draw it so nicely, the contours, and I didn't train it on that many regions of interest, but still it does, it didn't do that bad. So if I trained it better, if I took the effort, then I might get a good segmentation quality. And we are coming near the end of this session, as I see. So I guess we have covered what I wanted to mention about segmentation. And I think that I won't be starting any other bigger topic at this moment. So if you have any questions now, please ask. If there are no questions, I will